We live surrounded by microorganisms, which would rapidly cause our death were it not for the complex network of cellular activity known as the immune system. In this programme, we shall examine the discoveries relating to one part of it. Immunity is mediated by two interacting networks. Cells like this macrophage belong to the more primitive network, which mops up antigens such as bacteria or viruses, but can't distinguish between one antigen and another. We're concerned with a more highly developed network, the so-called adaptive immune system, which is mediated by white blood cells called lymphocytes. Each lymphocyte is able to recognize just one antigen out of the hundreds of millions that may enter the body. Experiments show that interactions occur between different types of lymphocyte, providing a complex system of checks and balances. Stem cells in the bone marrow give rise to two types of lymphocyte, T cells and B cells, which have different functions in the adaptive immune response. An antigen which enters the body triggers the adaptive immune system by stimulating T and B cells which are committed to respond to that antigen alone. When it activates a specific type of regulatory T cell called a helper, the helper in turn stimulates a B cell to start producing antibodies, also called immunoglobulin molecules. The B cell produces only one kind of antibody, specific to that particular antigen. The antibodies bind with the antigen, which facilitates their disposal by the body. When the antibody production is no longer required, another type of regulatory T cell, called a suppressor, switches off the B cell. So each B cell is part of a trio with a helper and a suppressor T cell, all responding to the same specific antigen. Yet another type of T cell, the cytotoxic T cell, has the ability to recognize specific antigen on the surface of both bacteria and infected body cells and destroy them directly. Embedded in all cell membranes are numerous proteins, carbohydrates and glycoproteins, some of which serve as labels or marker molecules. These marker molecules identify the type of cell this is, a macrophage or a liver cell or a B cell perhaps. Other molecules identify the species from which the cell comes. And there's also a set of molecules that are unique to the individual of the species. And these are called the Major Histocompatibility Complex, or MHC. And some of these MHC molecules appear on every cell in the body. Now, the MHC is as unique to an individual as a fingerprint. For example, it's certain that my MHC molecules are different from yours. And unless you have an identical twin, it's equally certain that your MHC molecules are different from those on the cells of every other living creature. Now, it turns out that the MHC molecules have a crucial role in the interaction of T cells with antigens, and we'll be coming back to that later. Now, all these surface molecules don't simply act as passive identification labels. They're also receptors, which can convey information into or out of the cell. So the obvious question is, how do they do that? How do they interact with other cells and molecules in the body? Well, more than 20 years ago, it was discovered that B cells interact with antigens by carrying embedded in their surface membrane a molecule of antibody, with the combining sites for antigens facing outwards. Antibodies are probably the best understood molecules in the immune system. With a molecular mass of 150,000, they are even large enough to be seen by an electron microscope. They appear to have approximately the shape of a letter Y. The backbone skeleton of the molecule bears out this general impression. The overall structure appears complicated, particularly when all atoms are displayed in a space-filling model, but both chemical and structural studies have revealed an underlying pattern. They show that the molecule displays a simple regularity. It is composed of 4 polypeptide chains connected by disulfide bonds. The long chains are known as heavy chains and the short ones as light chains. Both are made up of repeating units or domains. In the case of immunoglobulin G, or IgG, the most abundant class of antibody, there are 2 in each light chain and 4 in each heavy chain. The domains show a great deal of similarity, both in amino acid sequence and in 3-dimensional structure. Those which do not vary from molecule to molecule are called constant domains and make up the main part of the antibody. The others, which make up the tips of the Y, vary slightly for each specificity and are called variable domains. It is these variable domains, one from the light chain and one from the heavy, which contain the binding site for the antigen. They are folded into 2 grooved barrels, which lie side by side and form the framework of the binding site itself. The sides of the barrels are made up of strands of regular sheet structure with interconnecting loops. The inner walls of the light and heavy chains, shown here in red, make up the sides of the combining site cavity. Some of the loops which protrude from these beta sheet walls vary greatly in sequence and sometimes in shape from one antibody specificity to another. It is these hypervariable loops, 3 from the heavy chain and 3 from the light, which determine the shape and chemical nature of the binding site, and therefore the type of antigen it recognizes. It's easy to see what an economical and yet adaptable structure this is, entirely built from repeating constant subunits, with the variability necessary for antigen recognition confined to a relatively small part of the molecule. Now, the genes underlying this structure turned out to be far more interesting than anyone could have predicted. The genes that code for the constant domains are common to all B cells, but the variable domains have to be produced in more than 100 million different conformations in order to combine with all the antigens that we know exist. Now, there simply isn't enough DNA in the cell to have a single gene for each one of these different conformations. The diversity is achieved by rearranging a relatively small pool of genes. Originally, the DNA contains multiple alternative genes coding for the variable domains of the antibody molecule. The DNA coding for the antibody light chain contains about 300 different variable or V genes, each capable of coding for the variable regions, plus a few joining or J genes which also contribute to the structural variation. Further downstream come the constant domain or C genes. Early in development, each B cell selects a V gene at random, and the DNA which lies between the chosen V gene and the J genes is excised by enzymes and the DNA strand rejoined. The DNA sequence from the chosen V gene to the C gene is transcribed into messenger RNA. Next, enzymes trim the RNA even further by excising all but one of the sequences transcribed from the J genes. This leaves a unique combination of V and J gene transcripts. Finally, the messenger RNA is translated into a protein light chain with a unique variable domain. A similar process of gene rearrangement occurs in the heavy chain. The idea that genes could be rearranged to generate diversity within antibody molecules was a revolutionary one. It was first proposed by Susumu Tonegawa, now at the Massachusetts Institute of Technology. He recalls the conceptual shifts that had to be made before his ideas could be accepted.
Many biologists thought that genes are very static in the lifespan of individual. So the idea that the genes rearranges during the B cell development was against that dogma. That's one. The second is there is this another dogma in molecular biology or genetics, which says one gene encodes for one polypeptide chain. Okay? So the idea of the variable region and the constant region encoded by separate genes sounds like there are two genes for one polypeptide chain. Okay? So that was the second point, which were not necessarily accepted by mainstream of biologists. So our finding went against both of those. That's a conceptual difficulties. But once it was shown, since the experiment was so direct, experiment data was so direct, it was quickly accepted by many.